Hello everyone. In this video, we are going to discuss about the factors which affect an enzyme activity. Okay, or you can say factors which actually affect the rate of a chemical reaction. In previous video, we have started with unit 5 enzymes, and we have discussed about various properties of enzymes and the mechanism of enzyme action using lock and key hypothesis. Okay, so let's begin this video with factors affecting enzyme activity. Now, the general factors which actually affect enzyme activity are temperature, okay, pH, substrate concentration, and enzyme concentration. Now, out of these four factors, the first two factors, temperature and pH, are actually listed in your IGCSE syllabus. Rest two factors are not given in your IGCSE syllabus, but for the better understanding of this concept, you must know how substrate and enzyme concentration actually affects the chemical reaction, okay, how they affect the rate of a particular chemical reaction. Okay, so let's begin with temperature. Now, in the last video itself, I've told you that every enzyme works best, or you can say that every enzyme is actually most active at its optimum temperature. Okay, now for example, in case of human beings, the internal body temperature of human beings is 37 degrees Celsius, fine. But this temperature is little lower than optimum temperature of all the enzymes in a body. Okay, so in case of human beings, all enzymes, they work best at 40 degrees Celsius. Or you can say, they work best between 37 and 40 degrees Celsius. Okay, and when we talk about plants work best at 25 degrees Celsius, fine. So the optimum temperature for enzymes in human body is between 37 to 40 degrees Celsius. And in case of plants, it is 25 degrees Celsius. Now, if you look on this particular image, okay, if you look at this particular graph, the graph is plotted between temperature and the rate of reaction. Now, you are aware that this is x-axis, okay, and this one is y-axis, fine. So graph is plotted between temperature and the rate of reaction, fine. Now, how the change in temperature actually affects the rate of a chemical reaction? Just take example of an enzyme in human body, okay. Now, the scale you're aware, this is 0, okay, 20 degrees Celsius temperature, 40 degrees, 60 degree, and 70 degree, fine. Now, for example, if the temperature is 0 degrees Celsius, so what will happen? The rate of reaction was also 0. Now, as slowly as the temperature was increasing, just look at this, just a second. Just look at this laser pointer. Initially, both the rate of reaction as well as temperature is 0. Now, as the temperature is increasing, slowly the rate of reaction is also increasing. Okay, why this graph is going upward? Because as the temperature is increasing, rate of reaction is also increasing. And finally, what will happen? When the temperature reaches 40 degrees Celsius. Okay, at that particular time here, the rate of reaction is highest. Why? Because that is the optimum temperature of enzyme. And at this particular temperature, enzyme is in its most active form. Okay, enzyme works best at its optimum temperature. Okay, now after optimum temperature, what's happening? Now look at this graph very carefully. When temperature goes beyond 40, 40 is the optimum temperature, fine. Now when temperature goes beyond 40, what happens is the rate of reaction suddenly decreases. You can see the curve drops down. Okay, the curve drops down. Now I'm just describing the graph. In next slide, I will explain you the reason. Okay, in IGCSE paper, sometimes a question is asked, describe the graph and explain the graph. So there is a big difference between these two words, describe and explain. When we talk about describe the graph, you just have to look at the graph and write down the points or write down the changes which actually are taking place in graph. Okay, and in case of explain, you have to explain the reason behind these changes. So what's happening here is how to describe the graph. As temperature is increasing, the rate of reaction is also increasing. Okay, rate of reaction is highest at optimum temperature, which is 40 degrees Celsius. Okay, Why? Because enzyme is most active, or enzyme works best at its optimum temperature. And as temperature goes beyond 40, or you can say as temperature goes beyond the optimum temperature of enzyme, the rate of reaction decreases. Okay, the graph drops down suddenly. Fine. Okay, I hope till this it is clear to you guys. Next is explanation of this particular graph. Okay, why it happens. Okay, now see, at optimum temperature, what was the optimum temperature? The optimum temperature of yes. What was the optimum temperature? The optimum temperature of that particular enzyme given in previous graph was 40 degree Celsius. Okay. Now what happens below optimum temperature is when initially the temperature was 0 degree Celsius. Okay. Initially the temperature was 0 degree Celsius. Now as temperature increases, what happens is the kinetic energy of molecules increases. Okay. What happens is as temperature is increasing, the kinetic energy of molecules also increases. Now we are talking about which molecules? We are talking about enzyme and substrate. Okay, enzyme and substrate molecule. Fine. So as temperature increases, what happens? The kinetic energy of these two molecules, enzyme and substrate molecule, also increases. Fine. Now, what happens because of increase in kinetic energy? The frequency of collision between them increases. Okay, they will move, their kinetic energy is increasing, so they will move very fast, and as they will start moving faster, the chances of collision between them also increasing. Okay, the chances of collision between them also increases. Now as the collision increases, what will happen? The substrate molecule will get fixed into enzyme. Okay, in the active site of the enzyme. Okay, as the chances of collision increases due to increase in kinetic energy, hence the formation of enzyme substrate complex will also increase. Okay, now what happens below optimum temperature is the enzyme becomes inactive. The energy of enzyme and the substrate molecule is very low. They move very slowly and collides weakly. Now these points are very important as per your IGCSE exam, because it is asked in exam generally that below optimum temperature what happens to enzyme, and above optimum temperature what happens to enzyme. Okay, so below optimum temperature, remember, the enzyme remains inactive. Okay, the enzyme is not damaged, nothing happens to enzyme. The enzyme remains inactive below optimum temperature. Okay, the kinetic energy of this enzyme and substrate molecule is very low, and hence due to low kinetic energy, the chances of collision between them is also very less. Okay, they collide very less. Now, as the temperature increases, what happens? You can see here, as temperature increases, what happens? The kinetic energy of enzyme and substrate molecule increases. Definitely. Then they move fast, and frequency of collision also increases. Okay, and at optimum temperature, the enzyme is most active, and the rate of reaction is also very high. The rate of reaction will be at peak. Okay, at optimum temperature. So you should remember this, that as temperature increases, so there will be increase in kinetic energy of the molecules. Okay, and molecules include both enzyme as well as substrate molecule. Okay, and due to increase in kinetic energy, both enzyme as well as substrate molecule, they move fast, and the frequency of collision also increases. Okay, and enzyme is most active at optimum temperature, and hence the rate of reaction is also very high. Okay, for example, if this question is asked in exam for 4 marks, so you must include the point of kinetic energy, the point of collision, the point of moving fast. Okay, because these enzyme and substrate molecules, they start moving faster, hence the chances of collision increases between them. Okay, so you must include all these points, including enzyme is most active, plus the rate of reaction is very high at optimum temperature. Okay, now the very interesting point, above optimum temperature what happens? Okay, above optimum temperature. Okay, this is very important. Above optimum temperature, actually the term you have to remember is the enzyme gets denatured. Okay, this term is very important. I just forgot to mention it here. Okay, the enzyme gets denatured. Okay, it is denatured. Okay, the enzyme gets denatured. Now, if the question is asked for 1 mark, that what happens to enzyme above optimum temperature? So you can just write down that enzyme gets denatured. Okay, you will be awarded with 1 mark. Okay, sometimes the question is asked for 2 marks or 3 marks. So at that particular time, you have to write down that above optimum temperature, the enzyme gets denatured. The enzyme gets damaged completely. What is the reason for this damage? Actually, above optimum temperature, the shape of active site of enzyme changes. Okay. What is active site? In previous video, I have told you, active site is actually a portion in enzyme where the substrate molecule comes and get fit into it. Okay. So above optimum temperature, the active site shape changes. Okay. Now, as the shape of active site changes above optimum temperature, what will happen? The substrate molecule will not be able to fit into it. Okay. Hence, the enzyme cannot function. Okay. That's the reason, as enzyme is not able to function due to the change in shape of active site of enzyme, what will happen? The rate of reaction will drop suddenly. Okay. That's the reason from optimum temperature. That's the reason that from optimum temperature, the curve comes down. Okay. From optimum temperature, if for example this is optimum temperature, from optimum temperature the curve comes down like this. Why? Because the rate of reaction decreases due to change in active site of enzyme, change in shape of active site of enzyme. Okay, due to increase in the temperature. So remember these points very well, that above optimum temperature the enzyme gets denatured, enzyme gets damaged. Okay, enzyme cannot be used again. Why? Because due to high temperature, the shape of active site of enzyme changes. Due to this, the substrate molecule cannot come and cannot fit into the active site of an enzyme. Okay, so remember these points very well as per exam point of views: below optimum temperature, during optimum temperature, and above optimum temperature. Now the next factor which affects okay, this points I've included actually. Yes, active site is no longer complementary to the substrate molecule. Okay, the same thing, a substrate molecule cannot fit into active site. That means active site is no longer complementary to the substrate molecule. Fine. Hence enzyme gets denatured and hence the rate of reaction decreases. Now the next factor which affects rate of reaction is pH. Now in chemistry you have studied that the formula for pH is the full form of pH is power of hydrogen. Okay. In general, we can say that pH is actually a scale which measures how acidic or alkaline is a solution. Okay. Now, for example, if you have been provided with a solution, okay, any solution, and you have to check whether it is acidic or alkaline in nature, you can use different indicators. Okay, there are various methods to find it out. But how will you be able to identify? How will you be able to check that whether it is acidic or alkaline? That you can do by using the pH scale. Okay, so this image shows the pH scale. Okay, pH actually runs from 0 to 40. pH 7 represents neutral. Okay, pH 7 represents neutral. Okay, so the pH of normal water which we drink in our home is, or distilled water, pH of distilled water is 7. Above 7, all solutions are called base or alkaline. Okay, alkaline. And below 7, all solutions are acidic in nature. Okay, so 7 is neutral. Below 7, all solutions are acidic in nature, and above 7, all solutions are alkaline in nature. Okay, that is pH 8 to 14, and here pH 1 to 6 or 0 to 6. Okay. Now, like temperature, every enzyme is also having its own optimum pH. Okay. For example, when we talk about various enzymes in human body, pepsin is an enzyme. You have studied in the lower class in grade 7 that pepsin is an enzyme which is present in the stomach of all mammals. Okay, and this enzyme helps in the digestion of proteins. It breaks down protein molecules into amino acids. Now what happens is this pepsin is released in stomach. It is present in stomach, and you are aware that our stomach always produces gastric juice, and hydrochloric acid is a very important part of gastric juice. Okay, it's a very important composition of gastric juice. So this pepsin molecule or this pepsin enzyme works in acidic condition in the presence of hydrochloric acid. Okay, and the pH of hydrochloric acid is 1 to 2. Okay, it is between 1 or 2. Hence, pepsin enzyme works in acidic condition. Okay, pepsin enzyme always requires very low pH of 1 or 2. Okay, but when food passes from stomach to the next part of alimentary canal, that is duodenum. Duodenum is the first part of small intestine. Okay, so when food passes through duodenum from stomach, there is another enzyme which is known as trypsin. Okay, now in duodenum, the pH is between 7 to 8. Okay, because the pancreas releases a juice which is called pancreatic juice, and liver also releases a juice which is called bile juice. So what these juice does, they have they make the entire food alkaline. Okay, they changes the pH to 7 to 8. Okay, hence trypsin is the enzyme which works at the pH which works at the optimum pH for trypsin is 7 or 8, between 7 to 8. Okay. Now, in previous video, we have studied about the enzyme catalase. Catalase is a very important enzyme produced in or present in all the cells, all living cells. All living cells. Okay. Hydrogen peroxide, that is H2O2, is produced as a byproduct in many metabolic reactions. Okay, and you are aware that hydrogen peroxide is highly toxic for all the living cells. So what happens is the moment hydrogen peroxide is produced, immediately this enzyme catalase present in living cells will break down hydrogen peroxide into water and oxygen. Okay. Now you are aware that cell cytoplasm is around 70 to 80% made up of water, and the pH of water is 7, neutral. Okay. So catalase is the enzyme which actually works at pH 7 in neutral condition. Okay, so remember that pepsin enzyme in human beings works at pH 1 to 2 in acidic condition. Trypsin enzyme in human beings works at pH 7 to 8 in alkaline condition, and catalase enzyme in cell cytoplasm works at neutral condition. Okay, that is at pH 7. Now the graph here shows the rate of reaction and pH. That means how pH actually affects the rate of reaction. Okay, x-axis here is having pH and y-axis here is having rate of reaction. Okay. Now this first curve actually shows you the rate of reaction of for pepsin enzyme. Pepsin, I've told you, it is a protease enzyme present in stomach. Okay, pepsin, the optimum temperature of pepsin is between 1 to 2, or you can just say 2. Okay, so if you look at this particular graph here, this is the optimum temperature, because here the enzyme is most active. If you just my line, I'm not able to draw straight. If you just come down, you can make out that it's nearby 1. Okay, it's between 1 and 2. Okay, the enzyme is between 1 and 2. Sorry, the optimum pH is between 1 and 2. Okay, so as the pH increases, slowly the rate of reaction also increases. But after optimum pH, the rate of reaction goes down. Okay, it drops down. Fine. Similarly here, for other enzymes present in human body, okay, for most of the enzymes present in human body, that optimum pH lies between 6 to 8. Okay, it lies between 6 to 8 or 6 to 7, you can say 6 to 7. Okay, 6 to 8 actually. Now as pH increases, slowly the rate of reaction will also increase. Optimum temperature is this, between 6 to 8. And finally what happens? Beyond optimum temperature, again the rate of reaction decreases and the curve drops down. Okay. So I hope this effect of pH is also clear to you guys. Now what happens beyond optimum pH? Why the pH, why the increase in pH beyond optimum condition actually decreases the rate of reaction? The answer is similar to what we have studied in temperature. Okay, as the temperature goes beyond optimum condition, the shape of active site changes. Okay, hence the shape of active site no longer remains complementary to the shape of substrate molecule, isn't it? Similarly, in case of pH also, the change in pH, okay, if the pH goes below, if the pH goes above optimum condition, what will happen? The shape of active site will change permanently. Hence, the substrate molecule cannot go and fit into the active site due to the change in shape of active site of enzyme. Fine. As the shape of active site of enzyme changes, the active site do not remain complementary to the shape of substrate molecule. Hence, substrate molecule cannot go and fit into the enzyme molecule, and obviously the rate of reaction will decrease because no longer enzyme can work on substrate molecule and break them into products. Next, factors which actually affects rate of reaction is enzyme concentration and substrate concentration. Okay. Now remember, for both of them, actually both these factors are interrelated to each other. How? Because as the concentration of both enzyme and substrate will increase, the rate of reaction will also increase. Okay. But the condition is temperature and pH both should be optimum. For example, if both temperature and pH you have taken optimum. Okay, if you have taken optimum temperature and optimum pH, so at optimum temperature and optimum pH, if the concentration of enzyme and substrate molecule will increase, the rate of reaction will also increase. But for example, if you are only increasing the enzyme concentration and substrate concentration remains same, so what will happen? More and more enzymes are present. Okay, so more number of enzymes, they will work very fast and they will actually break down or they will actually make products out of every substrate molecule immediately. Okay, but in the later stages, what will happen? Enzymes are present, but substrate molecule concentration of substrate molecule is less. Hence, this becomes the limiting factor. Okay. Similarly, for example, if you have kept enzyme concentration constant and only the substrate concentration you are increasing, so what will happen? Enzymes will become less. Okay, so enzymes will become less, and lesser number of enzyme molecules immediately or very fast cannot digest cannot break down. Okay, cannot break down all these substrate molecules immediately. Okay, so what happens is, if we'll increase enzyme concentration and substrate concentration together, the rate of reaction will increase. And if you'll increase only one maybe enzyme or substrate concentration, so the another one becomes limiting factor, and the rate of reaction will increase only up to a point. For example, if you're increasing enzyme concentration, what will happen? Rate of reaction will increase only up to a certain point. That means the point till where substrate molecules are present. Okay, once substrate molecules get over, the rate of reaction will again drop down. And similarly, if substrate concentration is increased, so enzymes will break down these substrate molecules into product at slower rate. Okay, because substrate molecules are more and enzyme concentration is less. Okay, so it doesn't matter if substrate molecule increases or not. If enzyme concentration is less, what will happen? The rate of reaction will also be less. Okay, so they both are interrelated to each other, and if the concentration of one does not increases, it becomes limiting factor for the another factor. Okay, so only this much in this video. With this, we have actually completed the entire unit 5 enzymes. In next video, we will discuss some of the important past paper questions from IGCSE board exams. Okay, and the paper code is 0610. Take care till then. Bye-bye.